I first want to just thank ACMG for the invitation to share our latest work on using artificial intelligence to make predictions about nonsense mutations and missense mutations in the human genome. It's a real privilege to be here because as a geneticist, we always look to ACMG to provide the diagnosis guidelines, and I think it's really exciting to sort of contemplate what the opportunities may be to use AI in revising those guidelines going forward. And so this work is jointly done with Vasilis Entranos at UCSF and is led by one of my postdocs, Nadav Brandeis. All right, so here are my disclosures before we start. And so, my lab is primarily interested in the broader problem of assigning function to sequence. And most of the time, I think, as geneticists, we think a lot about common variants that are associated with common diseases. But in newborn screening and in many realms of human genetics, I think there's a lot of interest in looking at rare alleles that cause Mendelian-like diseases, or even those that don't really fit patterns of Mendelian inheritance, but are incredibly rare in the population. And so the work that we're presenting is really focused on trying to make some progress in functionalizing rare variants that might be segregating in the human population. And I'll say that there's a real role for doing this sort of independent of large-scale sequencing studies, and that's partly because of the structure of human populations, because we all came out of Africa and that there's a population bottleneck, that in many cases, rare alleles that are segregating in humans are so rare that it might be difficult to detect, more than just a few individuals or a family. So this problem of using computational predictions to functionalize rare variants, and in particular, these are going to be coding variants, is not a new one. It's been sort of pursued for at least a couple of decades. And the overall goal is really to try to predict whether or not a rare or somatic coding mutation is going to have some function. And function here is broadly defined. We can think about clinical applications, which is what this talk is going to be mostly focused on, whether or not a variant is going to be pathogenic or benign, or it could even, but it could also affect some biological function. If I have some time, I'll show some results of that as well. For example, whether or not a particular mutation is going to lead to a cellular phenotype that's predictable. There are quite a few variant functionalization approaches, computational methods that are out there, and most of them historically have been supervised. And what that means from a computational perspective is that the method is built and trained with some labels that already exist. And so this really started with SIFT in 2003 and Polyphen in the 2010s, and fairly recently CAD. And so, the idea is to use some of the latest and greatest at the time machine learning approaches to treat basically human or any really sequences as just a set of alphabet, and to try to build a computational algorithm that can predict whether or not a particular missense mutation is going to be pathogenic or benign. And these models will incorporate many different features, including specific changes from missense changes of different amino acid residues, for example, from a hydrophilic type of a residue to a hydrophobic one. It also incorporates in allele frequency information and evolutionary conservation. But more recently, and this is really happening as in some ways the consequence of having very large databases, genomic sequences, not just in humans, but across the domains of life, is to now build unsupervised methods that do not require any labels to try to predict the effects of variants. And so in 2021, Deborah Martz's lab published this paper describing Eve, which is really showcased how at the time, first-in-class performance of these unsupervised methods to predict the effects of missense mutations in the genome. And more recently, a method came out of Illumina called Primate AI that utilizes almost 1,000 genome sequences from closely related primate species to humans as a further filter to identify variants that might be pathogenic in humans. And again, all of these methods are not explicitly modeling evolutionary conservation. In some cases they are, but the computational sort of engine here is in many ways in learning implicitly evolutionary conservation. for those methods, and really in other realms of really our lives, is that the concept here is that if we have enough data, we can build models that can both predict labels, but also can generate new data that fit the labels. And so I think it's hard to now avoid what's happening. And I think, let me just touch a little bit what you can do in terms of utilizing these generative AIs. So, the models are based on this concept that really sort of mimics in some ways how our brain works, is that you would have multiple different, very large scale convolutional layers that sort of all feed into each other. And these sort of very complex machines, by taking in some input data, can build a computational model of that data. That model can then be interrogated to be able to make predictions, but can also generate new data. And so that's why they're called generative models. So for example, for imaging, this is in some ways a solved problem, but Google's been building these generative models. In 2012, you can predict cats from YouTube at 81% accuracy. And today, this is sort of a solved problem. But I think what's really exciting and really sort of captured the imagination of a lot of us is that these models can also be generative, starting with, this entirely new art movement called Inceptionism, creating what looks like, conceptually certainly interesting works of art, even if they're not artistically sort of, as captivating as some masterpieces through history. But I think more recently, OpenAI put out a model called DALL-E 2, where, really, like, if you just put in a text prompt, in this case, teddy bears mixing sparkling chemicals as mad scientists, steampunk, it produces an image that really sort of, looks like what the text was supposed to capture. This is also happening in biology, and most notably, AlphaFold, a model that's developed by Google, has transformed structural biology and is able to now predict protein structures at very, very high accuracy. So these deep learning models are a much broader class of models, and it can work on images, it can work on text, it can also work on music and other types of information. But specifically, I think for biological sequences, we're really interested in a particular class of such models that works on what we call sort of natural languages. And these models, and again, this sort of area of AI has existed for a long time, which is called natural language processing. And there are a lot of similarities between human languages and biological sequences. And so what is NLP? I thought it was kind of cool that we can actually go to ChatGPT now, which is a generative model, right? So it can predict sequences or text, but it can also generate new text given a prompt. And so here, one of my postdocs just asked ChatGPT to explain NLP to a child. Natural language processing is a way for computers to understand and process words and sentences that people use. And so, for example, you can tell a computer, I'm hungry, and it can use NLP to understand what you mean and respond in a helpful way. And so that's one thing you can do. But as many of you probably know, ChatGPT is really quite amazing. You can even go in and say, write a poem about a chatbot being asked to write a poem about a chatbot being asked to write a poem about infinite recursion, and it'll produce something that's somewhat sensible. You can also ask it fairly complex queries, like solve this, mathematical problem that's formulated in text, as well as, like actually write pieces of code that basically tries to accomplish a particular task specified here by a prompt, which is to animate the rocket ship horizontally, bouncing off the left and right walls. And so the core of all of these models, and there's going to be a lot of similarities to sort of like what we're working on, is this concept that, again, given enough information, maybe we can sort of train a model to learn that information without any labels. But how does this model learn without any labels? Well, that's not really true that it doesn't have any labels. It does have some goal that the model is trying to accomplish. And the goal is actually fairly simple. Given some original text, the model is just going to randomly mask out some of the words. And so the task then becomes design a model or learn a model that can predict with very high accuracy, which can be measured computationally, the missing words, right? And you can now train this model or refine the model computationally over and over again. And it's fairly computationally intensive to do this, but given enough notes, it can actually do quite a good job in predicting the missing text. And so this is really the concept behind ChatGPT as well as Lambda, which is Google's version of this. And so, maybe it's already sort of clear to you that biological sequences looks a lot like human text. It's just a string of letters from sort of a corpus and from an alphabet. And so here, for example, if we have the cat sat on the mat, we can tokenize the sentence by sort of the following. We can say that there's a start token, and that we can either tokenize it by each letter, or we can tokenize it by each word. And similarly, we can do that with a protein coding sequence. Whenever we see a methionine, we know that this is the start of a protein, and we can tokenize this either individually by every amino acid, or we can tokenize this by pairs of residues. And so, this lends, in many ways, sort of learning the dependencies of sequences in a protein sequence using the same sort of computational approach as we would use to learn the dependencies of either letters or words in human language. And so this is actually something that was built, but it wasn't built in academia. In fact, it was built by MetaAI, which is a branch of Meta, or also known as Facebook. And it's a protein language model that basically does exactly what I said, sort of using the same approach for the English language, but now for protein sequences. It ingests in all 250 million protein sequences that currently exist in UniProt across all domains of life. And then for each sequence, it basically goes in and randomly masks certain positions, and then trains a 650 million parameter neural network to try to predict the masked positions as high accuracy as possible. This was originally published by Alex Rivas in PNAS in 2021, and they've really shown that this approach, protein language model, which is called ESM1b, can be used and be very effective for many tasks related to studying protein sequences, one of which is in fact to predict protein structures. And so it has similar performance to AlphaFold. The outputs of this model, or what the model actually learns once it's ingested in all that data, can be sort of visualized as a substitution probability matrix. And so we're going to show some of these plots later on. So I just thought that I'll orient all of you. What I'm showing here is sort of a arbitrary protein that's about 36 or 35 amino acids long, and those are the columns. And each row is one of 20 amino acid residues that's possible. And so whenever you see sort of a colored box here, that means that this is the residue that's actually coding at a particular location. And if you do make a substitution, that's either going to result in a protein sequence that is not likely to exist in nature, or is likely to exist in nature. I think intuitively you would think that amino acid residues that have similar biophysical properties may actually result in fairly high probabilities for substitution, but those that are very different may result in a mutant protein that sort of would have very low likelihood. And so that's really our hypothesis, is that since the model, this ESM1b model, already learns the substitution probability matrix, maybe we can query it with mutations to the wild type protein coding sequence, and that damaging mutations would be predicted to be less likely. And what's nice is that this is an in silico model that's already pre-trained, so we don't have to retrain the model. We can just ask the model, what do you think is the probability of this new sequence that's got a mutation in it? And so that's exactly what we did. We took this ESM1b model, in many ways we sort of hacked it so that it can perform this very specific task where the model can spit out a particular score for the wild type sequence, and then we ask the model to spit out a different score with a single missense mutation in it, and then we calculate the ratio of that score, and those scores are probabilities, and so this ratio is log likelihood. And this being a pre-trained model, and that all we're doing is querying it, it's in fact extremely fast to go in and generate a score for every single missense mutation in the human genome. The other thing that we could do with this is, we don't have to put in, just one isoform at a time, but we can put in all possible or annotated isoforms with particular mutations in it, and that allows us to also identify isoform-specific effects, which I will discuss later on. If we just look at a few examples of this model learns. So here's, for example, 3 different genes. And again, this is just an actual data of what I described earlier of the substitution matrix, where the columns are each of the residues for each of these proteins, and the rows are one of the 20 amino acid residues. And you can see that places here that light up are regions where an individual substitution is going to result in a significant change in the likelihood. And so I hope we can sort of appreciate is that the regions that are lighting up are overlapping areas of multiple sequence alignment. So that's what the MSA coverage is. So that's not perhaps surprising, is that regions of the protein that's really important are going to be highly conserved across the domains of life. And so if you have a single substitution in that particular region that's missense, it could result in a mutant protein that looks really different from the wild type sequence. And that's what the model is essentially calculating, what this particular score is calculating. But what's also cool is that, this model, or ESM, and this is one of the advantages of ESM compared to other methods like Eve and primate AI, is that it doesn't actually require multiple sequence alignment. Right? There are many regions of a protein that can't be aligned reliably for various reasons. And so, for example, for this stastin gene, there's this MIT domain that doesn't have any MSA coverage, but is annotated as a domain. And again, here we can score that certain missense mutations will result in a very different sort of protein product. And so ESM is implicitly learning structure, like all these models are. And here, this is just a demonstration that the structure that's learned actually does align with, in these instances, known domains for these proteins, as well as areas of multiple sequence alignment. Okay, so that's the model. And that's what, we built this approach to basically score these variants. But is this score useful at all, right? This is just sort of like a computational prediction. You can sort of think about the score as a measure, right? If we talk about function, it's a sort of a measure of function for this particular protein. Is this mutation going to result in a wild type, or would it result in sort of a loss of function or non-functional version of this protein? So we're going to take that score and we're going to try to evaluate its ability to predict potentially more interesting phenotypes, like is a particular mutation going to cause disease, or would it be likely benign? And I think this is probably most relevant to this audience. So for this, we did 2 different sets of evaluations. We took ClinVar, which many of you probably are familiar with. It contains 1.5 million variants. 40% are annotated to be benign or likely benign. 8% are pathogenic. Interesting, 40% are variants of unknown significance, right? This is a big problem, is that we want to be able to annotate more VUSs, and then 12% are annotated as other. I'm an immunologist, and so many genes that we're interested in, unfortunately, are not in ClinVar for historical reasons. And so we also analyzed a different database called the HGMD, which is a private database that contains or attempts to collate all known gene lesions responsible for human inherited disease. And so in total, it's about 330,000 variants. So this database doesn't contain any benign variants. And so for the native set for that, what we took is a set of variants from 1,000 genomes, from Nomad that occurs at an allele frequency of greater than 0.1 or 0.5%. And so for both of these databases, I believe that most of the variants are annotated based on the latest ACMG recommended evidence and guidelines. So if we look at ClinVar, and so again, in ClinVar, they're both pathogenic and benign annotations. And then for our other evaluation, which is sort of look at the variants annotated in HGMD versus variants that are in Nomad at an allele frequency of greater than 1%, you can see that the single score that we're going to calculate basically creates a bimodal distribution. Right? The pathogenic variants in ClinVar, as well as the HGMD variants, they seem to have a very similar distribution. And then the Nomad variants and the benign variants have a different distribution. And based on this bimodality, we can sort of like almost draw a threshold fairly easily here around -7. Right? So if this log likelihood ratio score is less than -7, then variants can be annotated as pathogenic. And then if the score is greater than -7, then the variants can be annotated as benign. And here on the right, just illustrating how useful it is to actually look at these two different databases, because there's actually not a huge amount of overlap between HGMD and ClinVar. Okay, and so here's the performance of testing on ClinVar HGMD. ESM, is really first in class as measured by area under the curve of the ROC curve. And this is, whether or not we test all variants or compared to methods using a balanced data set where we only look at the variants that are annotated in these existing methods. And the method slightly beats out Eve, but again, it's sort of best performing even compared to some of these methods like SIFT and polyfin that basically has is trained on the labels. So, but more importantly, so I think if you look at the overall area under the curve, ESM and Eve behave similarly, but in a realm for clinical diagnosis, where you may want to sort of minimize your false positive rate, and let's say that we set the false positive rate at 0.05. Here ESM1b, again in ClinVar and HGMD, has significantly better performance than Eve in calling true positives, right? 60% versus 49%, and 61% versus 51% at a very low false positive rate. And so for clinical diagnosis, I think this realm might be much more interesting sort of than the entire ROC curve. So what are the clinical implications of this, right? What we've shown here is that based on known labels, the single score for ESM seems to be able to distinguish benign versus pathogenic variants. And you can see that if we expand it beyond just benign and pathogenic, the likely benign variants in ClinVar certainly look more like benign, and the likely pathogenic variants in ClinVar certainly look more like pathogenic. But now if you take the variants of uncertain significance, we can see that, at least based on the ESM score, they also actually look kind of bimodal. And about 58% of them we can annotate as benign, and the other 42% we can annotate as pathogenic using a Gaussian mixture model. So we're very excited about this. The idea here is that if you do have a patient walking into clinic and you have their exome sequenced and you have a variant of unknown significance, this is now a piece of evidence that now can be used to potentially prioritize variants. That's clinical predictions, but that's only, clinical outcomes is one phenotype, and it's one way of functionalizing variants. But, in many ways, it doesn't really tell us what the variants are going to do. And so, but there actually exists now quite a bit of data, and it's a growing collection of data where basic scientists are taking certain genes that might be of clinical relevance and performing biochemical assays where they're going to measure the effects of all of the possible missense variants for these genes using assays that we think are going to be relevant to the function of the gene organismally. And so if we take the set of existing what we call deep mutational scan data, or DMSs, there's a total of 16,000 variants across 11 genes that we could identify. And here again, ESM has first-in-class prediction. And this is now a quantitative prediction, right? We're taking that score, which is a continuum, and correlating that ESM prediction score with the measured biochemical assay score, which is also continuous. And you can see that in a rank-based test, this has an average correlation of upwards of 0.45, which is actually, we think, is quite remarkable, considering that this predictor doesn't know anything about the biochemistry or the molecular assay that's being performed, and doesn't know anything about the particular genes that these experiments are actually interrogating. And so here again beats out Eve and every other existing method that we could get our hands on. And so that's functionalizing individual variants. One of the, I think, cool again, properties of this in silico method is that we can really just put in any sequence we want. It's easiest, right, to start with the wild type sequence and make a single missense mutation, but we could also, in fact, put in indel sequences. And so it's not too hard to devise a alternate score. It's not exactly a likelihood ratio score. It's a sort of pseudo likelihood ratio score, which why there's a P in front of it, that basically computes a score where you can again calculate the difference or the likelihood of a mutant sequence. In this case, will be a small indel versus the wild type sequence. And so, again, we took this pseudo likelihood score and asked whether or not it can be useful in some way. So it turns out that there's not a lot of indels in existing databases. In ClinVar, we identified about 1,700 benign indels and about 1,800 pathogenic indels. And if you look at the distribution of the score, of the pseudo likelihood ratio score between pathogenic and benign indel variants, again, you can see sort of it's not as clean as the missensitive variants, but there's still, I think, pretty clear difference in the distributions. Again, if we sort of go back to that strategy of based on these two distributions, we were to draw a single threshold and evaluate the performance of that threshold as a classifier for variants that are pathogenic versus benign, ESM right here, and several different versions of calculating that pseudo likelihood ratio score sort of performs similarly at a area under the curve about 0.87. And compare that to other existing methods. And here, there really aren't that methods that can predict indels, but CAD is one of them that can predict the effects of indels. And so CAD has a AUC of about 0.84. And so ESM here significantly outperforms CAD. And then if you sort of use very simple approaches, like looking at distances, edit distances based on sequence or alignment, or maybe by actually performing a BLAST P first and look at the differences in the BLAST P scores, none of these methods do very well. They just are barely above random compared to ESM and CAD. So another sort of application of ESM and this model that we can interrogate, we can just sort of query is and flexibility of just really like inputting any sequence we want. Is putting in different isoforms of the same gene. And so you can imagine that, you have this gene that's got 4 different isoforms. And in this first exon, any mutation here would basically be pathogenic or damaging for this protein, and maybe pathogenic for a clinical phenotype that's related to this protein. But depending on which isoform it is, mutations in these middle exons could be either pathogenic in one isoform, but benign or neutral in a different isoform. And so it really allows us to at least ask the question of, are there variants in the human genome where a missense mutation may be pathogenic in one isoform context, but benign in a different one? And so we perform this assay or perform this analysis and identify, and here we're looking at about 3,500 ClinVar variants where there's a significant difference between a variant. There's a significant difference between the score of the variant on one isoform, or here we're calling the maximum isoform score, versus the score of the variant on another isoform or the minimum isoform score. Right, so here are a set of variants where it's essentially predicted to be likely damaging or pathogenic for one isoform, but neutral in a different isoform. So if we look at the set, again, if you look at the distributions here, if you look at the min isoform scores, many of them appear to overlap that distribution we've seen before for the pathogenic variants, which is so this ClinVar pathogenic. And then if you look at the max isoform scores, which is this, the filled plot here, that overlaps the ClinVar benign distribution quite well. And so we think that there do exist potentially variants where in the particular isoform context, it could be pathogenic, but benign in a different isoform context. And so that's quantified up here on top. You can in fact take the set of VUSs and show that for some of them they're potentially isoform specific. And so what are some of the properties of these isoform-specific variants? We certainly think that, right, one of the ways that a variant could have isoform-specific effects is if it, affected splicing directly, right? Could create an alternate splice acceptor site, for example. And if we take the set of isoform-sensitive variants and compare them to a random set of variants, we can see that, yes, it's true that the isoform-specific variants have much higher probability of disrupting a splice site. And that's both for a donor site as well as an acceptor site. And so let me just give you sort of one example of a gene called menin. And so here, what I'm showing on top are two different isoforms. There's the primary isoform, and then the second isoform that basically contains a 5 base pair deletion. Right? So this is a very, very small deletion in a region that if you look at the primary isoform, you go, well, this region doesn't really light up. So it may not even be a critical domain. It could just be sort of a hinge region of this protein. But that small deletion in isoform 2, you can see that when that deletion is there, you have this huge region right before that deletion that now lights up, right? And so in fact, you can take these two matrices and calculate a difference, a score difference. And so it turns out this region that in the primary isoform that may not be structurally important, in the secondary isoform, in the context of a 5 base deletion, could be structurally now much more important. And if we now sort of pull in 3-dimensional structural data about this protein, it turns out that it makes a lot of sense that in the context of isoform 2, where you have that deletion, there's basically this new pocket that gets a binding pocket that gets created, and that some of these residues that do not interact with each other in isoform 1 are in fact interacting with each other in 3-dimensional space. So again, these algorithms are implicitly learning, right, structural data, and that information, even though we don't actually have the structure every time, can be useful in predicting the variance, the effects of variance in a particular sequence context. And so just let me do a quick time check. So I want to end with 2 vignettes of where we're going with ESM next, which I think is quite exciting. And so, again, we're interested in functionalizing variants, and ESM is essentially learning what is an individual missense mutation going to do on the function of that protein. And here it's sort of measuring essentially structure, right? And we can show that that score can be useful in predicting organismal level function, whether or not a variant is pathogenic or benign. But what about everything in between? What about the actual biology, right? And so we're in immunology lab, and we're interested in particular, T and B cell signaling, but we're also interested in the function of many myeloid cell types. And so we sort of asked the question of, can these scores that's predicted by ESM actually, tell us something about the function, the biological function of these genes, using assays that we are sort of routinely running in a lab. And the particular gene that I want to show you an example of is this gene called ZAP70. And it's an important kinase that is really sort of turned on in response to proximal TCR signal. So that's when your T cells sees a antigen that gets presented by antigen-presenting cells in response to a viral or bacterial infection, or maybe some sort of nucleic acid damage that's created by cancer environment. And this gene has been sort of studied quite a bit, where different variants of the gene has been made in a mouse, for example. And so we thought, we can just take all missense mutations of ZAP70 and calculate, right, ask ESM to sort of give a score for this. One thing I want to sort of highlight here is that, what we've actually asked ESM to do in everything I've shown you before, is kind of, just taking the model and deriving a one-dimensional score, right? We're asking this model to say, give me a single number to predict whether or not a sequence is likely or not. So for the wild type sequence, it'll be high likelihood. For a mutant sequence, it'll be low likelihood. But the model implicitly actually learns something more. It learns a 1,000 dimensional embedding for each protein sequence. What that means is it's sort of like learning 1,000 features about the sequence. Those features, unfortunately, can't just be easily mapped to like features that we're used to thinking about when we think about proteins, like hydrophobicity, for example. It doesn't map neatly to that, but in some ways, it is capturing that biochemical information in 1,000 dimensional space. And so one thing we could do is, in fact, take that 1,000 dimensional embedding and try to visualize it in sort of 2 dimensions. And this is a common approach in computer vision and high dimensional analysis. And so what I'm showing here is, again, ZAP70. And here, each gray dot represents a missense mutation of ZAP70. And now what I've done is take that 1,000 dimensional embedding of ZAP70 and put it in 2 dimensions. And I've also now circled the wild type, which is right there. And so if you look at, in ClinVar, pathogenic versus benign variants of ZAP70, you can see that many of these variants, at least the pathogenic ones, are pretty far away from wild type. And so here, distance is somewhat arbitrary, but it is still somewhat correlated with that likelihood score that we calculate, which is also try to measure distance between any 2 protein sequences. So that's kind of encouraging to see. But then if you take another set of ZAP70 variants that's been functionalized by Art Weiss's lab here at UCSF, you can see that many of these islands, right, actually are enriched for variants that are known to be hypomorphs, which would be loss of function, as well as hypermorphs, which would be sort of gain of function for ZAP70. So it seems like in this high dimensional space where ESM is just learning implicitly about protein structure, maybe it's actually learning something beyond, right? Like in that high dimensional space, it might actually be able to distinguish variants that have a certain biological function versus a different set of variants. So we're really excited about, now the ability to go back to this model and ask it even more complex queries. But how does it do, like if we just want to convert that back into that 1-dimensional score, right? So again, ZAP70 is important for T cell activation. And so now we can do these, high throughput screens where in primary human T cells, we can go in and use a base editor to make endogenous C to T mutations. Now, we can't target every single missense mutation using this approach, but we can target quite a few. And we can take cells that are now phenotypically high for one particular molecular phenotype we're interested in, here, the expression of gamma interferon, and then take cells that are low and sort them, right? And then perform a statistical analysis. So this is not all that different from a GWAS, where you take individuals with disease versus those without and then sequence their genomes. But we've introduced the mutations in vitro, and this will be a class of mutations that might actually not be segregating very much in the human genome for reasons I sort of mentioned in the very beginning. And so, so we can now compare, right, this result here on the x-axis of this in vitro experimental assay. Each dot here represents a base editor's, a single base editing or a single missense mutation that's introduced by a base editor on the x-axis. And now we've measured the effect of that mutation using this high-low sorting strategy and calculating a log odds ratio. On the y-axis, we're showing, right, the predicted ESM1b score for each one of these mutations, again, for ZAP70. And so, we were pretty floored when we saw this result. Again, ESM, not knowing anything about the function of the gene or the biology that the gene is important for, is able to predict this experimental output at fairly high correlation. So the score isn't just telling us pathogenic versus benign, but that there's some, right, like the continuum of the score actually matters as well. And what's exciting is now if we go back to that embedding, right, we can in silico, using this computational approach, make predictions for every single missense mutation. And with the experimental approach with high throughput base editors, we can cover a lot, but we can't cover the whole set of possible missense mutations. And so we're excited about really the integration of these approaches. Imagine we have one of these islands where we know a lot of variants are clustering together. We can now go in with a base editor and functionalize a few of them. And maybe that'll give us information about all the other variants that are in that cluster. And I'll just end with one last example of another gene that we're pretty interested in called P2RY8. This is in collaboration with Jason Sister's lab here at UCSF. P2RY8, is a GPCR that's expressed on the cell surface, and it's been known to be important for B cell signaling, right? So if you have high if P2RY8 expression, you have low migration of B cells in the germinal center. So no P2RY expression, high migration. And variants of this gene recently has been described to be associated with sort of, more Mendelian or somatic versions of lupus. So again, we could sort of like do the screen. In this case, it's an overexpression screen. We're going to make a library of all P2RY8 mutations. And here it's actually a subset of that library of the ones that we have some prior information on. And then we transduce that library into cells that lack endogenous P2RY expression. And then we can do two different assays, right? We know that we can measure P2RY surface expression, but since we know that this is related, the function of the gene is related to the migration of cells, we can also do a transwell migration assay and actually measure the rates of migration of B cells, and again, sort for both of these phenotypes and perform a statistical analysis for each of the phenotypes. And so when we do this, right, we see this great, beautiful pattern here. On the left, we're looking at P2RY8 expression. And here, the y-axis is the output of that screen, and the x-axis is the predicted score from ESM. And so here, you can see that there's a set of variants where ESM predicts them to be loss of function from the perspective of the protein, and these result in no expression of the gene on the surface. And interestingly, right, these are correlated with increased B cell migration, just as we predict. So no P2RY expression, high B cell migration. So you see this nice correlation between P2RY8 expression and the ESM predictions of each variant, and then this nice inverse correlation between B cell migration and the predicted ESM scores. But here's an example, right, of if you now look at the expression of P2RY8 and B cell migration, again, you see that this nice inverse correlation. But here's an example of individual variant, right, where sort of these two assays are telling you different things. So here, the variant in ESM is predicted to be loss of function, less than -7. But if you look at the expression of P2RY8, it doesn't really change the expression of the gene very much, right? There's not a huge change in fold change. In fact, it seems to increase it. But now we go over to the B cell migration assay. Well, this gene definitely affects B cell migration, clustering with all these other loss of function genes. And if you sort of now look at this in the correlation plot between the P2RY8 screen and the B cell migration screen, you can see that it's one of the outliers where it doesn't lie nicely in this, x equals -y line. And so here, this I think is an instance where ESM is predicting something. This particular mutation will have an effect on the function of this protein, but doesn't result in its complete loss of expression on the cell surface. And again, now we're trying to analyze, right, this high-dimensional embedding of P2RY8 in ESM score space to understand if this is a mutation that's clustering in a different region from all the other mutations over here that are clustering together that results in loss of P2RY8 expression. So, this resource or these scores that we computed using this MetaAI model called ESM can be accessed by anybody. And so it's all pre-calculated for all 450 million missense mutations in the genome. So you can just, sort of look up any annotated isoform for a particular gene. There's about 40,000 isoforms, I think, in total in this database. And you can look up the score and download it and interrogate your favorite mutation or look at your favorite gene. And I highly encourage you to do that. And certainly reach out to us if the computational predictions do not align with sort of like what you expect to happen. And so just to summarize, I think we're not the first ones to show that, but this is sort of like one of the papers. And I think there are several that now are showing the same thing, which is that protein language models are now first in class in their ability to predict pathogenicity versus benignness. But these models are incredibly powerful in the sense that they're learning implicitly protein structure, and they can go beyond just predicting ClinVar, HGMD, NOMAD. But just for that particular task, it predicts at a AUC of greater than 91%, which is first in class. And I've shown that some of the applications of of the model that goes beyond just predicting the effects of individual missense mutation is that we can predict isoform-specific effects as well as effects of small indels. And we also have some evidence internally that we can predict the effects of fusion proteins. And beyond a single label, the model, what the model learns can also predict the outputs quantitatively in a continuum of deep mutational scanning data, as well as these sort of high throughput base editor and transgene overexpression assays. And so with that, I'll leave my acknowledgements up and just acknowledge again Nadav, who's the postdoc in my lab, who did all the work. And this is a collaboration with Vasilis at UCSF as well. And since the publication of the paper and just sort of my summary of how we think this work can contribute to the progress of rare disease research, we've had lots of different people actually, and this is in some ways the most rewarding part of it, reach out to us who've got, either individual patients or case studies that they've been working on, and they have a variant of unknown significance where they can now look it up in this database and at least have some, score that they could, interpret as distance from this wild type protein. And in some cases, it is a isoform-specific effect, which is why it's sort of been missed previously. And so we're pretty excited to continue to work with those groups to at least study their individual proteins, and through that process, really understand, what might be some of the limitations of the method, right? Like, I think intuitively we can think that if you are looking at a gene that doesn't have a lot of conservation, maybe the method isn't going to make the most meaningful predictions. But we really think that it's just another tool in the toolkit to be able to score many of these variants that will hopefully be identified through exome sequencing studies. And what's nice about the work is that, it can provide a score essentially for every single missense mutation, which I don't think a lot of the tools can do. And we're happy to continue to work with the community to better understand sort of, again, what do these scores actually mean. And so with that, I will take any questions live. Thank you.